We're going to move on to our next speaker, Dr. Amber Goetz. She's a principal investigator in the North America Product Safety Science Strategy pillar of Syngenta. She received her Ph.D. from North Carolina State University and then joined the product safety at Syngenta. And much of her effort has been to advance risk assessment paradigms and supports the development of novel science technologies to influence positive regulatory changes in North America. Dr. Goetz will be the first of 3 speakers in our session now on successful application of NAMs for regulatory purposes. And the title of her presentation is Implementing Modern Approaches to Testing and Evaluation into Weight of Evidence Assessments.
All right, thank you, Norm. Thank you, everyone. Thanks for this invitation to come and speak. I've learned so much talking with several folks over here in the past day, so I'm really looking forward to this afternoon's conversations. So I'm going to do a quick test here to make sure I understood the remote control functions here. All right. Yes, okay, we're winning. All right, so what I'd like to do here is, so I'm from Syngenta. So it's Syngenta Crop Protection, and yes, I work with agrochemicals, pesticides, plant protection products. So I work for Syngenta, agrochem company, and what we've been doing, and it is very happy to report that with the presentations we've had today has really laid a lot of great groundwork for what we're working on as well. Using these decision trees, developing these frameworks to improve our safety testing, our paradigm that we use. So what I'd like to do here today is show you a few examples of what we've been using at Syngenta, and I believe several others are as well, as we've seen from some of the examples today, and looking into those 3 R's. How can we reduce, refine, and improve reduce the vertebrate testing that we use for our safety assessment. So again, keep in mind 20 minutes, so I'm going to keep an eye on the watch here so we can keep the flow going this afternoon. But I'll start off with what are the knowledge needs when we are working with our agrochemicals, and we want to build these new approach methods into our assessments, but we need to build that internal as well as external confidence in using these. And then I'll discuss a little bit on how we are using the weight of evidence approach and, again, incorporating those new approaches. And with the time available, I'll share as many examples as I can. So the key message here, if we want to stop right now, is using the key knowledge accumulated from your intended use, i.e. what are those application rates? What is that exposure? What are we looking at? What's your exposure? And what do you know about this molecule in particular, this classic chemistry? Get as much information as you can to address those questions for safety up front, and that can really tailor your science plan of where do I need to focus my attention first. If this is a mutagenic compound, I'm not interested. That's a given. But if I know that this classic chemistry tends to have an effect on the kidneys, for example, again, that's where I'm going to focus my attention right away. If this is not an acceptable risk, I'm not going to continue with this chemical. Okay. So we're not going to talk about all of these risk knowledge needs. What I wanted to share with you is there's a lot of questions that you need to assess when you're looking at a pesticide. I'm just going to focus here on the human health risk assessment. And what you'll see here is that you need to look at it from the duration of exposure as well as the amount of exposure, of course. Some acute, intermediate exposure, long-term exposure, all routes of entry, oral, dermal, inhalation, and the list goes on. Of course, you do not want to have a carcinogenic, mutagenic, or reproductive toxicant on your hands. So these are the numbers and types of questions that we start off with. Here's your new compound, Amber. Make sure that it doesn't have any of these effects. What am I going to do first to begin assessing this to identify, yes, this is an ideal candidate to go forward with? So you want to look at I'm not going to talk through this slide. We do have a PDF to share if folks are interested. But yes, let's identify and characterize that hazard. Let's look at those exposure and identify those target organs of toxicity and what those effects are. So by doing that, we want to reduce the animal testing, and we've identified some new approach methods. I'm very big on weight of evidence. I'm very big on using read across. Let's learn as much as we can about this chemical and structurally similar compounds. And that's all well and good, but in order to get this acceptance, if you will, building this confidence in using these new approach methods, what I'm happy to say here is getting in touch with your regulatory environment, with the reviewers at the EPA Health Effects Division as early as possible to start having these discussions. I'd like to approach this from a different level. This is how I'd like to address it, and have those conversations, identify what those key questions and concerns are so that we can work on this science plan together and build that development. So if you can characterize that exposure, you can characterize those effects, but I don't need to do a cancer bioassay, okay, let's talk. Let's find a better way to use that with the approaches that we have today. So I'm going to spend just a few minutes here. I'm pretty sure everyone in this room is very familiar with the weight of evidence, but just to get you onto the same page as the way I think about weight of evidence, and I have to say the Hall et al. Paper does a very nice job on this, so I've adapted a few figures from that paper just to share. So when you're thinking about it, these lines of evidence, is it a qualitative? Is it corroborative? Is it quantitative? Is this approach method going to really address your question of concern? And where does it fall in that axes of reliability and relevance? By that, I mean reliability, what's the quality of that data? Is that a reproducible protocol? What's that data? Is that something that I can reproduce and have a lot of confidence in? Yes, it's reliable, it's good quality, I can use it. What's that relevance? When I'm looking at this weight of evidence as an entire body, how much weight, if you will, should I put on this line of evidence? Is this more mechanistic? Is this more corroborative? And again, is this more towards the human exposure? So with that, you're looking at variable lines of evidence here. I'm just expanding on the same concept here. And so again, from left to right, in vitro, my favorite, toxicogenomics, metabolomics, ex vivo. If you've done a study and you want to do some additional work, keep those samples, utilize as much so you don't have to repeat that study. And then again, you move into metabolomics. You have your pharmacokinetics and understand what those metabolites are. And again, you just work your way towards what is that relevance. So lastly on this, just to, oops, try it one more time. There we go. So here's just a quick example of an in vitro assay. Yes, it's very relevant. It's very reliable. This is good. However, this is more mechanistic. Does this really, it does help to support and address the question that I have, but let's add some more information on that. Let's look at our in vivo transcriptomics. We've just recently started working with in vitro transcriptomics, so that expands the chemical space with which we can work. And again, with transcriptomics, you can identify the mode of action. You can also exclude modes of action. So you can really help to tailor that science plan, that safety assessment package. And then, of course, read across. Let's pick a class of chemistry here. Let's say we're working with triazoles. I'm going to look at as many triazoles that are structurally similar, have biologically similar, have the same pesticide mode of action. Not all triazoles are the same. What are those most similar compounds that I can treat as my chemical analogs for that read across? So again, as many lines of evidence, as much available data as you can to build up. So that's where I am when I'm thinking of weight of evidence. And what I'd like to do here now is just to showcase several of the work. Some of them have already been raised earlier today, so this will be all the more opportunity to put them in the limelight. And you'll see there on the lower right is something, and I'm sure several folks here, Alina Sinswinski, put together a very nice paper on problem formulation. Before you get started, what are the goals and objectives of your assessment? What is that question? What is the problem statement? The problem is I don't know when I might be able to waive this study type because I don't have the criteria and the guidance defined to say, yes, I can identify that point of departure. Yes, I can conduct that risk assessment without that study. That's just an example. So using that problem formulation really helps us to get that conceptual framework so that we can then move forward and conduct our weight of evidence assessment. And I'm happy to report that with this new approach, we really have gotten a bit more efficient, if you will, on screening chemistries as we're working on new classes of chemistry. Using these hypothesis-based inputs and the read across and identifying, we don't want to work with an HPPD inhibitor if we don't have to. We don't want to work with this type of chemistry, so we're going to weed out and screen earlier on. So that's been very helpful for us. Okay, so looking at this, I'm going to try to go through at least 2 examples here. The first of which is one of very close to my heart. Transcriptomics was where I did my graduate research. So what we've done here over the past several years is making more use of our early stage research. This is, a lot of people don't like to hear about this, but when you start assessing the safety of a compound, what's that dose range finder? What's that maximum tolerated dose? How high do I go before I see an effect? All right, now we're going to dose them a little bit longer. What's that maximum tolerated dose? So you really work to refine that dose. Can we cut all that out? We've known that we've been doing this for quite some time. And if we take a look back over the past 10 years, just to start with, we rarely go above 300 mg per kg, and we will see those effects as sufficient. We're not going to test in a long-term study greater than 300, in some cases 500 mg per kg. So I don't need to go to that limit dose. Let's bring it back down. Okay, jump into, do your short-term study, but do a little bit more with that first short-term study. So we start with the 14-day, we do the toxicokinetics, we look at the any apical endpoints, any effects that are observed, but we've also included transcriptomics, metabolomics, and we also included pathology, of course, as much information out of that 14-day. And as I mentioned, you can identify as well as exclude modes of action. If I'm not seeing a peroxisome proliferator, great, I don't want to worry about that. If I'm not seeing something that's activating the aryl hydrocarbon receptor, again, great, I've excluded those modes of action. If I'm seeing an effect in the kidney or perhaps the thyroid, okay, you need to start thinking about liver-mediated mode of action to assess what is that a direct or indirect effect on that thyroid. So again, it steers your science strategy. What types of studies am I going to do based on this information? And you can get a lot out of 14 days, provided one of our personal criteria is that you have to reach a steady state exposure. And that usually can happen within the first 5 to 7 days. So a 2-week study is sufficient so that we can see those effects. So when we look at that, this is something that I'm very happy to share, is that we really shortened that time frame to making those key decisions in our early stage research. It can take several years. Now we've really reduced that down to less than a year to identify, yes, this is not useful compounds. We're not going to see these effects. We can forecast, predict those long-term effects. And likewise, we can predict that registration success, that we're not going to have to deal with these effects. So again, it's a very useful tool for us. This is, it's not used for registration dossiers per se. However, it is very useful for us to understand more about our compound before we go too far. So again, this is just to share a slide, all the types of data that we are pulling out of our short-term studies. And by doing this, we have stopped doing some of our other subchronic studies. We don't need those. We have enough information to go straight into the long-term studies. So that's one example. Keeping everybody on time here today. This one is, I'm happy to say, Rama's paper. This was shared. Dr. Marshall did a very nice job, and I couldn't agree more. This is using the source to outcome approach, and being what's the source, what's the exposure, what is the dosimetry, and what is that outcome. We've used, as Dr. Marshall mentioned, the mucilare. What are those effects? Best part of that, we're using human cell lines. We don't need to do an extrapolation, so we know what those effects are. Again, let's start with the source. What is that particle size distribution of this pesticide? What is that inhalable fraction? What is that respirable fraction? All right, let's take a look at the dosimetry. So what is that, if you're looking at the application of this pesticide in these products, is this a drench application where you're just drenching the soil, or is this a backpack spray, so you're gonna have fine mist? What is that exposure? What is that likelihood? Dosimetry. What is that deposition? Back to the fluidics model, how much, and this is where we can begin to compare the difference. What is that deposition in the human airway as well as in the rat airway? And then finally, the outcome. Using this modeling, using this refinement, and this in silico approach, we've been able to use this to say, here's the human equivalent exposure. This is the dose level. This is the risk. We've been able to identify the point of departure and successfully use that in our risk assessment. So again, instead of doing a 28 or 90-day study in a rodent, we've been able to actually use human-specific to identify those endpoints. And this will be my last one, just to wrap it up, is this is what is my personal favorites, is that we're working on. We've actually gotten this to the IATA stage. We've just submitted this proposal for an integrated approach to testing and assessment method. And what we want to do here is, in addition to refining and reducing, in this case, I am working on a weight of evidence framework where we can advocate that we do not need to conduct a cancer bioassay. That's a big step. How are you going to demonstrate that? What do you need to do to show that you have more than enough information here? And this is just to, as I said, this is in the proposal stage, so this isn't published. Fingers crossed, we'll be saying different news this time next year. But this IATA, this framework is a process. It's a weight of evidence framework to identify that point of departure from the subchronic studies. So we haven't replaced, we haven't taken the studies out yet, but this is, if you will, the baby step, getting from where we are today with the traditional approach to moving away from animal testing to using in vitro. So the first step is, do we have sufficient information? Dr. McGurney, she did her paper, and this is one part that the whole paper is great. But looking at that decision tree, you have all these multiple iterative processes within that decision tree. I would love to tuck this one in there as well. This iterative process is, what do I know about my compound, and what do I know about structurally similar compounds? Again, from their structure, from their biological effects, from their pesticidal mode of action, what is that confidence that I can use that chemical analog to read across and get more insight into mine? Likewise, what is that necessary information that I need? If it's genotoxic or if it's mutagenic, from an ag chem perspective, we're not interested. But that's not the case for industrial, for others. So again, we would like to expand this workflow to other types. Moving on down, as again, looking at all of your short-term and subchronic studies, do you see any pre-neoplastic lesions? Maybe you have some omics data. Do you see any effects? Can you identify what that mode of action is? The better you understand that mechanism of action, the better you understand what that biological response is. And if you have that health protective threshold at which no effects occur, you now have a protective point of departure. You do not need to do as higher for longer term. You've identified when there is absolutely no biological effect. Likewise, on the left-hand side of the screen are those read-across chemicals, asking those similar questions. But what I find very useful in this exercise is, in some cases, picking on triazoles today, but in some cases, you might have another triazole that doesn't have those effects, or maybe it has effects that you didn't see in your compound. That's okay. It doesn't necessarily mean it's an unreliable read across. What that tells me is I need to take a closer look. Did I see any such effects? Pick your favorite organ. Did I see any effects in the kidney? The data over there is take that closer look. The big point with this weight of evidence assessment is not an absolute guarantee that you don't have to do the study. What this weight of evidence is it identifies for us what are those key knowledge needs. Do I have any data gaps? And if so, then I need to go and I can do a tailored alternative approach, smaller, well-designed study to answer that question. I don't necessarily need to conduct a 2 or 3 year study to answer that question. So those are the key lines of evidence, I think, for carcinogenicity assessment. We can make use of in vitro and short-term assays to evaluate our carcinogenic potential. And we can, the advantages, of course, here is that we can really set those exposure limits to prevent those primary effects and protect against all adverse long-term effects. So I think I'm going to end with this slide to give time for the rest of the speakers this afternoon. But this is just several folks are probably familiar with the Risk 21 approach. And what we've done is just on the left-hand side, you can see all the sources of data that we used in this weight of evidence assessment. And by using our no AEL, or no observed adverse effect level from our 90-day study, we did pass the risk assessment. It was well below the level of concern by the US EPA, and it was a higher NOEL than our actual. We did actually do the cancer bioassay. It was lower, but despite that, we were able to use the subchronic pass risk assessment and still get the use patterns that we wanted. So those are just a few examples that we use here at Syngenta. So I'd like to pause there, see if there's any questions. Yes, please.
On the last slide, you had indicated that you have a lower, NOEL value for the tox study compared to the subtox study. Did I hear you correctly?
Yes, so we had used, let me see if I said this correctly. So our 90-day, we used 15 mg per kg, and our cancer bioassay was 4.7 mg per kg. So we went ahead and we used the subchronic 15. We used the 100x uncertainty safety factor for intraspecies and for intra. However, we also added, because this is new, what is that confidence in using a subchronic study for chronic dietary risk assessment? So we added an additional 10x. So this is 15 mg per kg with a 1000x safety factor. Whereas if we had done the cancer bioassay, we would have done a 4.7 mg per kg with a 100x safety factor. So it was a smaller number, but we're still able to use it. Yes. And no, you're absolutely. Yes, it is very conservative, is a nice way of putting it. And when you think about that, as I mentioned earlier, with the internal confidence and acceptance of this approach, and that's where, again, this is in the beginning stages, this is a draft proposal, but that's where we're headed with this, saying, yes, it's a lower endpoint, because you can imagine, well, now you're reducing your risk cup. I'm not gonna be able to get as many crops in there, I'm not gonna get as many uses in there. So are we sure we want to do that, or are we going to go forward and do our 2-year multi-million-dollar study to do that? And so those are the types of questions that need to come into play when you're making that call. Yes?
Can I just have one more follow-up? I know we're short of time.
Yeah.
Have you looked at that also, the changes of the certainty factors in terms of subchronic to chronic, in terms of S share, some of our papers, in terms of short-term to chronic, and instead of a full 10x?
I will look that up. Thank you for that. And where I'm headed, where we're talking right now is, okay, we are doing PBK modeling for quite a number of things. So could we factor in our kinetics and say, yeah, we can reduce that by another 3x? So thank you for that suggestion. I'd probably move on. All right, thank you. Yeah, happy to follow up afterwards.
Yeah, we'll have time at the panel discussion to follow up if we need to. Yeah.
All right.
Thank you so much.
Thank you.